All right, interruption, and I'm afraid that I can't figure out how to fuse videos, but regardless. We're continuing on with DNA as a transforming factor. So, the idea is that if you take the virulent bacteria, heat them up, you kill them, they're dead, and nothing happens to the mouse. We can pet this mouse too. Good little mousey, you survived. Now, what Avery did, is if he took the live, non-virulent bacteria and mixed it with the heat-killed virulent type, and you actually had to do something that rendered the bacteria what he referred to as being competent, which is actually adding a little bit of calcium chloride. Then, all of a sudden, you would inject the mice, they would die, and you would get virulent type S bacteria recovered. What he has discovered is something he calls the transforming principle, in that there is something released by S when you heat them up that is transforming R into a virulent form. Now, of course, we know what he's doing is heating is popping the bacteria cells open. The DNA can get taken up by the non-virulent cells. They take that DNA up and they are now going to be lethal. But he didn't know that. What he calls this the transforming factor. And this is referred to as the transforming principle. All right, so now the hunt is on. What is the transforming principle? Now, or this transforming factor? So back then, they had, biochemists had some fairly crude tools to look at different biomolecules. One of the tools was they could take enzymes that would specifically degrade proteins. So if they take the heat-killed stew, the mix, and they would add proteases to it that would break down all the proteins, well, it turns out that they still had virulent bacteria being recovered. So that throws out the idea pretty well that proteins are the transforming factor. If they take enzymes that break down carbohydrates like starches, that doesn't do anything. It turns out if they take enzymes that break down RNases, that still didn't stop the transformation. But when they took an enzyme that will break down DNA, a DNase, all of a sudden they no longer have the avirulent bacteria becoming virulent. That is the first point, that DNA may be the transforming factor. So people began to look at DNA in a more careful light. And a guy named Chargaff, and some of his work was actually being done at U of I back in the 30s and 40s, Chargaff was able to break apart nucleic acid polymers and begin to actually quantify the amount of individual nucleotides. And what he discovered was really pretty interesting, is that the amount of adenine was equivalent to the amount of thymine, so A equals T, and the amount of guanine was equivalent to the cytosine. This became known as Chargaff's rules. And what he was able to demonstrate is that the AT to GC ratio is typically constant for species, within a species, but it can vary between species. And what he is determining here is genome content, genome complexity, all kinds of stuff. But this is another really cool idea now we have to fit into the mix. Somehow A is equivalent to T in number and G is equivalent to C. Now, just as a little spoiler alert, and if you knew this back in the 40s, you could win your very own Nobel Prize. That's because we had the double helix where A base pairs with T, so that's why the AT numbers are identical, and G base pairs with C. Here's your Nobel Prize. Thank you very much. All right. So, more experiments were being carried out. And a really cool experiment carried out in Cold Spring Harbor by Hershey and Chase. Chase, by the way, is a woman that was doing this work, which is pretty cool in the 40s. The Hershey-Chase experiment was making use of a virus that infects bacteria, something that's called a bacteriophage. And the mechanism that bacteriophage are able to infect bacteria wasn't fully known. But what they did was made a very special bacteriophage where they labeled the proteins on the phage with a radioactive sulfur, S35, which is part of methionine, one of the amino acids, and not at all part of nucleic acids. And then they radioactively labeled DNA with radioactive phosphorus. And there's no phosphorus in the proteins, particularly in the outer covering of the bacteriophage. So they had these two discrete radioactive labels, S35 methionine for the protein and P32 phosphorus for the DNA. And what they were able to do is, by taking sulfur-labeled bacteriophage, infecting bacteria, so the bacteriophage kind of lands on the surface of the bacteria and injects nucleic acid. They then whiz this thing up in a blender, and that is knocking the bacteriophage off the surface. And it turns out that when you do that, there is no radioactive sulfur inside the cell or in the cell pellet. All the radioactive sulfur is in solution, which is where the protein shells are for the phage. The next experiment they did was the real clincher, where they've got the DNA labeled, the P32 labeled DNA. Same thing happens, you get infection, it dumps the DNA in, they whiz it up in the blender, knocks the bacteriophage off, spin down the cell pellet, and lo and behold, radioactive phosphorus is now in the bacteria, and there's nothing in the solution. They have discovered that DNA is being injected into the cell, and that is what's causing the phage to be able to be replicated. They have pretty much defined the fact that DNA is the genetic material. That is a really cool, big discovery. So it's then left to people to start figuring out the structure of DNA. We know that we have the AT interaction, and this is X-ray diffraction that was generated by Rosalind Franklin and Wilkins, where, I mean, to me, this looks like either a Rorschach picture or some sort of symbol for the X-Men, or Avengers a symbol. No, whatever. But they are able to determine from this photo that DNA is helical, that it has a width of about 20 angstroms, which is about that big. No, whatever. It has a repeat unit in that they can see it swiveling around, and then there is a very narrow repeat unit as well. When you begin to put this data together, the best model comes from it being a double helix. Think of it as an extension ladder, an aluminum ladder that a giant comes up to and goes and twists. So you've got the places where you put your feet on the ladder. That's actually the AT or GC hydrogen bonding. And the rails of the ladder coming up the side, that's the phosphate sugar backbone. And there are a couple of competing models, but the best one that fit all the data is, of course, what's known as the Watson-Crick model, in which Linus Pauling and others had proposed a model in which the nucleotide bases were sticking out, because that's actually the only difference there. And so they were thinking maybe that was a way that the nucleotide bases were being read. It didn't fit with the model very well, and only when they actually had the bases go in did this thing actually work. And we have Watson and Crick and others developing this model known as the double helix. So this is our extension ladder. We have the sugar phosphate backbone, and then the nucleotides, a TA-AT base pair and a GC base pair here. And that makes sense. It fits with everything. The replication of this actually begins to make sense as well. So the Watson and Crick model, the AT-GC ratio is fixed in a species. This fits with Chargaff's rules. You got the space between the bases and the space between the twists that fits the model that Franklin and Wilkins put together. You don't have anything that is going to interfere with developing the order. Hydrogen bonding is what is binding the AT-GC base pairs, and that can be broken fairly easily. Boiling will do it. And they, in this quote, or in the paper from Nature in 1953, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So, when you very carefully lyse a bacteria cell and place it into an electron microscope with super high resolution, we see this massive amount of string that is the DNA. And then you see circles of plasmids, and here they are in a higher mag, kind of colorized. So we've got, in most bacteria, a really big chromosome, between 0.5 to 5 million bases, megabases, and plasmids that are much smaller, generally between 1,000 bases or so up to 200,000 bases. All right, we're going to stop here and pick up on DNA replication on Friday and see where we go from there. Thank you for your attention, and I'm Dr. G. I'm out of here. Peace.